Hello. I'm going to start today's session by acknowledging the traditional custodians of the land upon which this festival is being presented, the Yagara people. I also pay respect to Australia's First Nations, who we recognise as the first scientists and custodians of the lands and environments and all animate and inanimate objects within it. I acknowledge the importance of their legacy, passed from one generation to the next, over 50,000 years, has on caring on Mother Earth and contemporary scientific conversations. I acknowledge the deep and timeless relationship between country and Aboriginal peoples and Torres Strait Islanders, and also pay respect to the elders of the community, both Indigenous and non-Indigenous, who are with us today. Good afternoon, ladies and gentlemen, and welcome to Pandemic, the Race for a Cure. Queensland Museum is proud to host the World Science Festival Brisbane, and we thank our partners for their support. I'm Tegan Taylor, a health reporter at the ABC, but more importantly are our expert guests today. Our first speaker is Dr. Larissa Labson, who is dedicated to unravelling the mystery of how our immune system detects and responds to acute viral infections, including COVID-19. Hi. And our second speaker is Professor Paul Young, who studies viral diseases and develops new and improved vaccines for viruses of both human and animal origin.
Hi Tegan, hi everyone.
Hi. Hands up if you've noticed that we're living through a pandemic at the moment. Let's talk about that for a bit. So, Paul, we know we're living through one, but what exactly is a pandemic?
Well, probably easiest to start with the definition of the word. I mean, it comes from two Greek words, pan and demos. Pan meaning all, and demos meaning people. Democracy comes from the same derivation. So a pandemic is essentially a disease that is impacting everyone across the globe. So you've probably heard of epidemics as well. Epidemics tend to be more localised, perhaps in single city environments or a country. Whereas a pandemic is a disease that can spread globally.
And so when you've got a pandemic, you've got three ingredients. There's the germ itself, it's got to be able to spread, and you need to have people who've never had it before so that it can continue to spread easily. So Larissa, how often does a pandemic happen?
Yeah, it's a really good question. We know that they've happened all throughout history. They're referred to sort of as plagues or general sort of Black Death, for instance. Big ones like this that we're aware of, probably the biggest one before this was the 1918 flu, which you might have heard of, killed about 50 million people. But as we've known in recent years, we're having more and more pandemics. So we had SARS in 2003, we've had Ebola recently, we've had Zika. So there's also the chance that they're going to keep happening more and more often as climate change becomes more of a phenomenon that really affects our lives. And this is because the more we interact and encroach upon wildlife habitats, just by sort of spreading urbanisation, the more we're going to be coming in contact with different animals. And these different animals harbour different viruses and bacteria, and so the chance for them to infect us, and we've never seen them before, so they spread, becomes much greater. So unfortunately, I think that we're going to be seeing more pandemics in the future.
So, oh yeah, go.
Yeah, maybe just to expand on that a little bit too. When we talk about viruses, certainly most people in the community, and indeed probably most scientists, just think of those that cause disease. You know, virus equals disease. That's actually not the case. Viruses are essentially just replicating pieces of nucleic acid, and they adapt to whatever environment they're in. So all hosts have co-evolved with viruses for millennia. And we carry our own viruses right now that don't cause disease, that you won't know about. We study some of them, but they don't cause disease. They sometimes have assisted us in our own evolution pathway, but they are essentially either have null effect or have beneficial effect to their host. So this co-evolution tends to move more towards that stable relationship between a virus and a host. When we see viruses emerging causing diseases, it's almost universally because the virus has shifted, spilled over from its natural host into humans or into animals. We sometimes see the same situation occurring in animal species, particularly domesticated animals. So this is when you've got a virus and a host that are not used to each other, co-replicating and each of them trying to adapt. You know, the viruses are being selected for the most efficiently replicating, and sometimes that is a situation that causes disease if that host hasn't worked out a strategy to actually combat it. So just remember that viruses don't always equal disease, but they're the ones we tend to talk about, particularly in the concept of plagues and pandemics.
So we're going to talk about viruses in a bit more detail in a minute, but let's start by talking about some previous pandemics, and let's start with one that's not caused by a virus, the plague. What was the plague, Paul?
Yeah, and as Larissa mentioned, plague tended to be a term used for disease that was being spread on a large scale, and so we've had multiple plagues over the years. The big one that we know from history, called the bubonic plague, was in medieval times. It was around 1350, and it actually arose as the spread of a bacteria called Yersinia pestis. This was a bacteria that was transmitted efficiently between animal hosts and humans via a flea, and the bacteria would infect the flea, and the flea would transmit it. This was a plague that was moving around China and India, causing devastation there, moving across into the Middle East, and the pinpoint date, it was, I think, October 1346.
Put it in your calendars.
Put it on the calendar. It was 12 ships that left the Black Sea and moored in Sicily, Messina in Sicily. And when those on the harbour came down to the boats, they discovered there were a lot of dead sailors on board and also many sick sailors. They knew about disease even in ancient Greek times, and you know the word quarantine comes from a very old use of the word. Quarantine, actually, the derivation is 40 days. And it used to be that they would quarantine ships for 40 days in harbour, just so if there was any disease on board, it would burn itself out. So they pushed these boats back out into the harbour, but it was too late. And that initiated a plague that then spread across Europe, and we think resulted in the death of about 200 million individuals. And in the 1300s, that was half the world's population at that time, so huge impact. But we've seen these plagues over many years. Historically, we know about the one in 165 AD, when Marcus Aurelius was the emperor of Rome. This was a disease that started in Rome and spread out, the Antonian plague, and then there was the Justinian plague that started in Constantinople and spread through Europe. That also had a huge impact in Europe at the time, killing something like 20% of the world's population. So we're continuously seeing these plagues over all of human history, and we will continue to see them beyond this one as well.
So Yersinia pestis isn't extinct, it still exists in the world, but we have ways of treating it now. But another famous pandemic that we saw more recently, like only 100 years ago, was the 1918 influenza, which we think of as the Spanish flu sometimes. Larissa, what happened then a century ago?
Yeah, so I think you've made a really good point. You know, Yersinia pestis is a bacteria, and a lot of those plagues, if they emerge now, we have better tools to fight them. We have antibiotics, and antibiotic resistance is a thing. But, you know, still antibiotics work pretty well. But with viruses, it's a different story. We don't have lots of equivalent antivirals that we can tackle those easily. So the Spanish flu, as its name suggests, is caused by influenza virus. And this came about in about 1918, so the same time as World War I. And it actually killed far more people than World War I, about 50 million is the estimate. And what happened was, as Paul alluded to, really this influenza, which typically infects birds, but also seasonally infects humans, basically crossed over from a previous bird or potentially animal host, so like a pig or a horse, and started infecting humans. And because we didn't have the right pre-existing immunity, it ripped through the population. And basically people were getting really, really sick with it. They couldn't do anything to really treat it except for nurse people. It was highly contagious, kind of like COVID is now. It's a respiratory virus, so you sneeze, you catch it. They were wearing sort of rudimentary masks. But essentially, you know, trying to shut things down and maintain social distancing, and that was all they could really do. And it basically went on to circulate. You know, a lot of us have remnants of the Spanish flu in the kind of flu viruses we have today. It's evolved on.
we'll come back to that in a second, because one of the viruses we're going to talk about, I think it's partially descended from Spanish flu. But first, what about smallpox? That's another big one that we sort of hear about in history books, but it's not a problem anymore.
Yeah, so smallpox is also caused by a virus. All of us are, well, I'm too young, and probably all of you are definitely too young to
Paul's
have been...
old enough.
Paul, you're definitely... Paul Young is... were you vaccinated against smallpox?
No.
No, so you see... So smallpox used to also cause a massive plague. Apparently, it was called the smallpox in contrast to syphilis, which was the great pox.
Wasn't that great?
And smallpox was given that name because of the characteristic blisters that it caused all over the skin. And so they became really pustular. So it was incredibly disfiguring. If you caught smallpox, you had about a 30% chance of dying. It was incredibly contagious. A huge number of historical figures had it. It was estimated about 500 million cases in the 100 years before it was eradicated. So smallpox is one of the only viruses we've managed to eradicate. So there are no cases of smallpox anymore. It was eradicated, I think you said, in 1977.
Declared eradicated then.
Yeah, so which is pretty incredible, and that is through the use of a vaccine, which we'll talk about soon. But essentially, it shows you just how powerful vaccines can be, that you can take this incredibly disfiguring and debilitating disease and you can eradicate it because you've managed to stop the virus from being able to replicate.
And it just shows the power of what we can do in terms of vaccination. There was an 11-year WHO concerted campaign to eradicate smallpox, and that relied on the delivery of vaccines in sort of ring-based approaches. So they'd go to where there was an outbreak, ring vaccinate around the population to prevent it from spreading out. The reason I'm certainly old enough to have been on the planet when smallpox was still here, but by the time I was born in Australia, we'd pretty much eradicated smallpox as a problem here in Australia, and that's why many of us didn't get vaccinated. Still some who travelled overseas would get vaccinated, but now we've got probably an almost completely susceptible population again. But it's no longer around. It's no longer around.
You've got an interesting historical factoid of someone who we know had smallpox in ancient Egypt.
Oh, well, smallpox has been around for a long time. We know that it was a problem in ancient Egypt, and one of the main reasons is that we discovered on Ramesses V mummy, not his real mummy, but his mummified remains, on his face were the telltale vesicular marks. So these aren't often when you have this disease, the repair of those pox become a mark and invagination, and so you see a sort of pock mark as a result of where the vesicle was. But this was a real vesicle still there, so we're pretty confident that he probably died as a consequence of smallpox. People about 15 or so years ago actually went in and were able to extract DNA out of that pustule and sequenced it and showed that it really was pox virus. So they were sequencing a more than 4,000-year-old piece of nucleic acid, which turned out to be rather similar to the smallpox that was moving around the globe in the 20th century. So that's a virus that didn't really change much over time, but it's been one of the scourges over many centuries up until recently.
So while we're continuing our terrifying trip down memory lane of all these different viruses that have affected humans, or pandemics that have affected humans, can we talk about some more recent pandemics, Paul? The swine flu is one, and Ebola.
Yeah. So swine flu, let me just go back to the comment that we made that pandemics and emerging viral and bacterial diseases are a common and consistent threat. Over the last 30 or 40 years, we've been seeing an increase in the number of emerging viruses and bacteria, not necessarily at a pandemic scale that we've now seen COVID-19, but in local epidemics. Larissa mentioned a few of them, Zika and SARS and so on. But swine flu in 2009 was an emergence of a new influenza that arose out of, yes, the animal population, but in an unusual way. It actually recombined. A number of different flu viruses got together in the same cell. There was a recombination event that led to a completely new virus that the human population had never seen before. I mean, it acquired components of a bird virus, basically. And when that suddenly made its way into the human population, there was no one with any immunity to it. And as a respiratory pathogens are notoriously transmissible, particularly influenza. Very rapid turnaround between infection and ability, and replication and then ability to transmit to another individual. And so that ran through the global population within a year. We were very fortunate with that one. We dodged a bit of a bullet. The expectation was that it would have higher virulency, cause more disease. It didn't. I mean, it still killed more people that year than flu normally does, so it was a major threat, but just not quite the equivalent of the 1918, 1919 flu that we were all fearing. More recently, there was the Ebola outbreak, and that's a really good example of how human behaviour changes the way the virus responds in a population. So we've known about Ebola since the 1970s, when it was first identified in Sierra Leone as a new disease. We've been getting sporadic Ebola outbreaks over the years, but they've always been contained within villages. Very high fatality rate of something like 80%. So unfortunately, it tended to wipe out a village very quickly and limit the ability of itself to transmit. This is actually a virus that's also in bats and possibly transmitted to humans via primates. But with Ebola, it tended to be contained within these village environments. What made 2014 different is that an infected individual made their way to the capital city of Guinea and started a human-to-human transmission that wasn't contained for over a year. It spread not only through Guinea but also into Sierra Leone and other countries in the region, causing a massive death. In fact, it was the single reason why the WHO got so much criticism for being reactive to that response. They were a bit delayed in responding, and they didn't really have much in their armoury to deal with it, other than to send out people into the field and try and deal with individual infections as they appeared. And they committed after the Ebola outbreak to be better prepared. The globe needed to be better prepared for the next big epidemic. And so between 2016 and 2020, there's been a lot of work in the background to make us better prepared. And to be perfectly honest, the reason why we've landed in such a good place, having recognised that this good place is following a year of real tragedy in terms of the impact globally, but we've landed in a place where we're actually rolling out a vaccine now in very short measure. So we learnt a lot in those few years about how we need to be prepared.
And one of the things that your team was doing in the background there, Paul, was figuring out ways of making vaccines that could be adapted to new pathogens that we hadn't seen before. So let's talk a bit about vaccines. Larissa, how do they actually work?
Yeah, so vaccines are really predicated on this idea of immune memory. So I'm an immunologist. I study the immune system, so I'm pretty excited about it. And basically, our immune system has a number of arms. It has an innate arm that recognises that there is danger, so that there's an infection and it poses a danger to the normal function of the body. And that will kind of sound like a local bell and will really wake up the rest of the immune system, which we call the adaptive immune system. And these produce antibodies, which I like to think of as kind of handcuffs that stop the virus or the bacteria from being able to get inside our cells, which they need to do in order to replicate. And then we also have another arm called the T cells, which will come along and kill any infected cells. So if you can imagine a virus or a bacteria-infected cell becomes like a factory for that virus or bacteria. So these are really important components to all fight off the virus or the infection. And so they take a lot of energy for the body to make up, and they also take time. So usually it takes about 2 weeks for your antibody and your T cell responses to kick in. And so often that lag time is long enough that the virus is able to replicate kind of out of control and it's too late. But it's kind of a situation of what doesn't kill you makes you stronger. So if your immune system does manage to get it under control and clear the virus, it keeps that information of how it made those antibodies specific for that virus or those T cells, it keeps that on file so that next time if you are exposed, infected by that virus again, you know how to fight it off. So this is called immune memory, and it tends to be why a reinfection is less dangerous than the first infection. And that's basically what vaccines do, is they're trying to trick the immune system into thinking that they've seen the virus before. And there are different ways that we go about doing that, which I think we'll talk about now, and some of that can be to give a weakened version of the virus, to give an inactivated version of the virus, so you're presenting all the bits of the virus to the immune system. You can give just a bit of the virus to the immune system, so a lot of the vaccines for COVID now are essentially doing just that. They're just giving one part of the virus to the immune system, and it's got to be enough that the virus can make a response to it, and that response is the right kind of one to be able to block the viral infection.
So let's talk about the first vaccines, Paul. Smallpox is usually thought to be the first vaccine, is that right?
Yeah, it's traditionally thought to be the first vaccine, yes.
Tell us about it.
The origin of what we probably call modern vaccines goes back to a long time ago, the late 1700s, when Edward Jenner made the observation, based on observations that were in the field at the time, recognised at the time, but made the observation specifically that milkmaids had a very fair complexion on their faces, but tended to have pox marks on their hands. And just remember, this is at a time when pretty much everybody in the population would have encountered smallpox. Even the king was known to have a pock-marked face. And so there was this thing ravaged through the populations. It would kill a significant number, and those who survived would have had this memory or these marks of past infection. Milkmaids didn't have that. And his observation that allowed him to develop the vaccine was the recognition that on their hands were these typical vesicles, but they were very similar to the vesicles that were seen on cows, particularly on udders in cows. And so as a milkmaid would be milking the cow, she'd be getting infected with these particular pathogens. And the recognition that there was protection against subsequent smallpox was the radical idea that Edward Jenner had. So his idea was to take cowpox and use it as a way of preventing infection in a particular individual. And he did the remarkable thing of carrying out this statistically irrelevant experiment of one individual and didn't pass it by his local ethics committee because he chose the son of his gardener to vaccinate with some of this cowpox material. And 4 weeks later challenged him with live smallpox, and he survived.
Thankfully.
Maybe vaccines would have come along some other way, but they came along because he survived. In fact, he went on to challenge little Mr. Phipps some 20-odd times and showed that he was protected each time. He must have had an amazingly boosted immune response by then. But remember, this is at a time when people hadn't really identified what a virus was. They knew about disease. They didn't know what immunology was. So it was really all observational, that exposure to a disease protected you if you didn't die. And that was the observation over many centuries. But the key observation was, okay, this is something slightly different, but still affords protection. Up until that point in time, there had been a strategy called variolation, which the Chinese had done more than 2,000 years ago, had been practicing more than 2,000 years ago. Smallpox would ravage through the communities in China, just like they would in Europe, and have been doing so for a long period of time. What they recognised, again, was that people who'd encountered smallpox and survived were protected from subsequent rounds of infections. So they thought, well, maybe if we expose those individuals to a controlled amount of the smallpox, maybe that would protect them. And they found that that was the case. They used to take pustular material from one of those vesicles, dry it down, make it into a powder, and blow it up the nose of recipients. So scabs going up your nose.
Scabs up your nose, that's great.
And the consequences of that there would be some people who would die, but at a population level there would be protection. So that was adopted and used over many centuries, and that practice was taken into the Middle East, and during the Middle Ages there was a lot of variolation practice carried on there. Europe knew about it, but this association with occasional deaths was something that they weren't prepared to go forward with, and it was really with Jenner's observation that you could use a related virus or a related disease context that didn't cause any disease, didn't cause death, that led to the modern concept of vaccines.
Did people like the idea of smallpox vaccines at first? Was it an easy sell?
No, there were anti-vaxxers even in those days. And the concept of taking, a scraping from one species and injecting it, what they did is they used a very sharp knife and would scarify the skin and inoculate that way, and the virus would grow in the outer skin layers and induce the immune response that way. And you can see from this cartoon from the London Illustrated News, the cartoonist has drawn little cow's heads coming out of people's arms because of this concept that they were messing with nature and that they were going to cause great damage. But in fact, what they did was, this was the start of eradicating a major population scourge from the planet.
And so in addition to fighting smallpox, that really kind of set the tone for vaccines for other diseases, what about polio?
Yeah, polio has been around for a long time. I mean, ancient Egypt comes into this quite regularly. You can actually find reliefs on some of the ancient Egyptian monuments where there is representation of individuals actually with a crutch and a wasted limb, that's clearly a result of flaccid paralysis, and that's one of the major symptoms, quite diagnostic of polio infection. Polio is a gut infection, and in most individuals it just maybe would lead to some diarrhoea, but not much more. But occasionally the virus can travel across the basement membrane lining the gut and hit nerve tissue and travel back along nerves into the central nervous system and cause paralysis. And certainly here in Brisbane, just like many places in the 1930s through to 1950s, we had whole wards of Royal Brisbane Hospital dedicated to individuals who'd been paralysed by poliovirus. So you may have heard of iron lungs. And essentially these were large drum-like machines that individuals would lie in, and they were actually providing the mechanical intubation for those individuals to allow them to breathe, because they had lost the muscle capacity to for their own diaphragms to drive lung expansion and collapse. So polio has been a scourge for a long time, and again, with the lessons over 100 years or more of what Jenner had done, there were a couple of approaches towards polio vaccination. One was they killed the virus, so used it, as Larissa was saying, it's about tricking the immune system into thinking the live virus is there. So if you kill the virus so it doesn't replicate but still looks like the same thing, you can stimulate an immune response, or you can weaken it. In the case of Jenner, he found an alternate virus that was not the same, but what we do in modern times is we weaken it in the laboratory so that it still grows but it doesn't cause disease. Both of those types of vaccine were established, and in the 1950s they were rolled out. The oral polio vaccine, which is the live vaccine, became the most dominant one, was rolled out right across the globe. And the WHO, like smallpox, had a pathway through to eradication, and they had their deadline of the year 2000. And by 2000, it had been eradicated through most countries around the world. We'd eradicated it in Australia, a long time before 2000, which is why we stopped vaccinating against polio. But unfortunately, in a few small areas of the globe, we still have the virus ticking over in the hundreds, even as we speak. But the reality is, this is a virus that's almost like smallpox, on the verge of eradication because of a vaccination use.
So thanks to vaccines, you guys have never had smallpox vaccination. You wouldn't have been vaccinated against polio either if you grew up in Australia. But there's lots of vaccines that you will have received. I mean, you get lots of vaccines when you're a kid. You would have gotten the measles, mumps, rubella vaccines. Maybe you've had your human papillomavirus, HPV vaccines as well. So that's sort of like the vaccine backdrop. But now we've got this new disease that we're dealing with. Larissa, what is COVID and where did it come from?
Yeah, so COVID is the name of the disease caused by the virus. So the virus is SARS coronavirus 2. So SARS stands for Severe Acute Respiratory Syndrome, which is a mouthful, but it really reflects that you have a lot of problems in your lungs and breathing when you have this virus. And that's because the virus gets into the cells that line your lungs and mediate gas exchange. So they allow you to breathe, right? Take in oxygen, breathe out carbon dioxide. And the virus infects them. And what happens is that as those cells get really sick, they essentially die, and you'll either get holes in the lungs, so you get a whole heap of fluid from your body going into your lungs and you suffocate, and also you're not able to breathe in anymore. So this is why a lot of people are on mechanical ventilation when they're really sick with severe COVID. So this virus It's an envelope virus, so I like to, as I explained to my friends, it's it's your nucleic acids, which Paul mentioned. So it's an RNA virus. It's a very long RNA virus, and it's encapsulated with a protein and lipid shell. And on the outside, it has this spike protein, which is what uses to essentially attach and get into the cells. And it binds on humans to this receptor called ACE2. So as Paul will talk about, a lot of the vaccine approaches are really around this spike protein. So what we know is that this virus, essentially, we think it came from bats. And why do we think that? Because original SARS, we think, came from bats, and many very similar viruses have been isolated from bats in recent times. So based on looking at the sequences, we really think that this is probably a virus that came from bats. What we don't know is how it got from the bats into us. Did it go through an intermediate animal? Was it able to get directly across? There's the possibility that there have been multiple crossover events, so it's gotten into humans quite a few times, but never been able to go from human to human. And that's what's really important for a pandemic to start. So whenever that happened, that's really when this whole thing has kicked off. And we still don't have the exact estimates, but of course the first cases were really reported in China.
So we were saying before, Paul, that, say, Ebola virus, at least the early outbreaks, were killing like 80% of people who are infected. COVID doesn't kill nearly that many people who are infected. So what is it that makes it so dangerous?
Exactly that. I mean, it's a combination of issues, that it doesn't have a high kill rate, but it has a high transmission rate. So it's able to be transmitted with respiratory droplets and more recognised now also aerosols. That combination of transmission and not as high a mortality rate, I don't want to underplay that, because it's a mortality rate that far exceeds what we see with influenza. So this is a serious infection, particularly if you're dealing with a virus that's spreading around the globe and through the majority of the population. We've now seen something like 2.5 million deaths. So we're talking about a large number of individuals who have paid the price for catching this particular disease. So it has sort of, I don't like to put it too much in these terms, but it has hit, from an evolutionary perspective, the sweet spot of transmission and mortality that allows it to transmit so efficiently around the globe.
So I was just going to say, another thing that makes it particularly annoying is that it's able to transmit before it shows symptoms. So it's either able to transmit when without people showing any symptoms at all, so there are asymptomatic cases, but also pre-symptomatic. So if you've been exposed to COVID and caught it, then quite possibly you can be transmitting that on to other people before you even realise you're sick and potentially get tested or whatever. And that's made it incredibly hard to control. And this is where we think it might actually be a bit different to the original SARS, SARS-1, where that was really sort of at its most transmissible when you were highly symptomatic. And as Paul has kind of said, this is why, original SARS potentially died off, even though it was far more dangerous, is because essentially by the time people were symptomatic, they were in hospital and they weren't able to spread as much. Whereas with this virus, people are out spreading before they even realise they're sick. So it's really this evolutionary trade-off for a virus.
And in terms of the severe disease being seen in the elderly and not as much in the younger cohort, and I should point out that there is plenty of real disease happening in the younger cohort, so it's not a virus you want to catch. But from a population perspective, there is a lot of asymptomatic infection, and that has allowed it to spread more efficiently as well.
Right, so you've got it spreading without people having symptoms or before people have got symptoms. And of course, up until very recently, we didn't have very much in the way of protection against it. So what are some of the tools that we have to stop the spread of a germ that we don't have a cure for?
Yeah, so we've seen a lot of public health interventions. So we were all locked down last year and also at the beginning of this year for those 3 days. We've obviously not been able to travel overseas. We've not been able to travel interstate. So we've really been trying to stop the movement of people because, of course, that causes transmission. You've heard a lot about social distancing, so maintaining that distance, staying far enough away so that you can't catch the virus in the first place. Good hygiene has been really important. But in the cases of people where you can't avoid catching it, so this might be primary healthcare workers, other situations where it's impossible to kind of control, we now have some more interventions that we really didn't have at the beginning of this pandemic. So, you know, when this first started this time last year, if you caught COVID and you were hospitalised, like, there's not much a doctor can do. So what we have now, we have monoclonal antibodies. So I talked about the immune system making antibodies. Well, this is basically scientists in a lab having identified what are really good antibodies and making them in the lab and then injecting them into people so they work to stop the virus from getting into cells. Former President Trump got that when he was sick with COVID. We have antivirals, which work in a similar way to antibiotics. If you think they come in and stop replication. A couple were tested for COVID and plenty are in development, but none of them have worked really well. And another thing that we've noticed is that really when people are sick enough that they go into hospital with COVID and they need to have mechanical ventilation, it's no longer actually the virus that's necessarily causing all the symptoms and the disease. It's actually an overactive immune system. So essentially the immune system is trying to fight this virus that it can't control, and it's caused a whole lot of collateral damage. So if you can imagine, instead of potentially the virus causing the damage to your lung cells, it's instead the immune system which is trying to take out those sort of viral factories, and instead is taking out normal cells as well. And so also the body is not able to repair itself quickly enough. So what they've actually found in severe patients who have been hospitalised, that some immunosuppressants are actually working well to kind of bring people back. So from my perspective, looking at the immune response and how we can really balance that and get that right is another sort of way to try and tackle this virus. But ultimately, you know, don't catch COVID, but prevention is better than cure, and that's where a vaccine, of course, would be great, and that's sort of where Paul comes in.
Right, but before we get there, what would happen if we couldn't, I mean, we've got vaccines for COVID now, there's actually quite a few of them, but let's say we're not talking about COVID, what happens if you don't have a cure for something like this?
You would stay locked down forever, or eventually succumb to a high level infection rate that would ultimately lead to a herd immunity where the majority of the population that survives have encountered it. I mean, we've seen the history in the past of what happens with plague and the rest when you don't have good public health measures and when you don't have appropriate cure strategies or prevention strategies. So the short answer is we would see a lot more disease out there in the community. I can't see us being locked down forever, so that would ultimately come to approach us. I mean, it's interesting. You know, we've done an amazing job here in Australia of curtailing the majority of the infection, but the irony there is we've done such a good job. We are probably the most receptive population to COVID of any in the world, because hardly any of us have had it.
That's unsettling to think about.
But it's not too unsettling, because we do have those vaccines in the background, and that's what's going to bring this curve down and open up the borders again. I might want to just quickly touch on a bystander effect of what we've been doing over this last year. And that is the observation we all made that there was no flu last year. It flatlined. I mean, when I say no flu, I mean no flu. We have many, many thousands of cases every year, and we can predict, which season we see it, and it just flatlined from March last year. So it just shows how effective. I mean, not that we want to be locked down forever, because that's one of the effective ways of actually locking this down. But certainly all of those measures like good hygiene and, maybe that's the end of the handshake. Maybe it's the elbow nudge from now on. It's that ability to prevent that physical contact on a regular basis that moves viruses around the community. So maybe something to keep in the back of your mind. Good hand hygiene from now on is probably a good strategy.
So as well as stopping COVID, those measures that we've taken that you talked about have prevented lots of diseases from spreading. So let's talk about vaccines. And so, Paul, your team at UQ has been developing a vaccine technology. There's other vaccines that have been approved for use in Australia. What goes into developing a vaccine from the start?
Yeah, probably a lot more work than appears on the surface. I mean, our particular vaccine was on the end of my own group's interest in vaccines for 40 years, and the development of underlying technologies and capacity to look at these. So there's a lot of discovery science in the background. but also about 10 years of specific work on the platform technology that we've applied to COVID. So there's a lot of background work to develop an approach where we can make that spike protein that Larissa talked about on the surface of the virus. Instead of the whole virus, killing the virus or making it a weakened version of it as a vaccine, such as smallpox was and measles is, we just take the gene that encodes that outer spike glycoprotein and make it in the lab. We make it in flasks in the lab, but we translated that to working with CSL, a manufacturer down in Melbourne, to work with huge fermenters where it was made in large scale. And we were literally making grams of this material that could be used for vaccination.
Sorry, the large scale is measured in grams, is it?
Well, large, very large, because what we do in the lab is micrograms, or even nanograms. So a gram and approaching a kilo is a lot of protein.
Okay.
So making a recombinant protein that you can show to the immune system, and it will generate immune response. But there are multiple ways of looking at vaccines now, other than just killing and attenuating. There's taking an innocuous virus, one that doesn't cause disease, and impairing it even more, but then putting the gene for that particular protein in that. And it becomes a delivery vehicle. It comes like a Trojan horse. So it can take the gene into a cell once it's injected and drive the expression of that protein so that the immune response is stimulated. And that's the AstraZeneca vaccine that's now being rolled out in Australia. It's delivered via a changed chimp adenovirus. So it's another animal virus that no longer replicates but can drive expression. Probably the most exciting thing out of the last year has been the coming to fruition of about 20 years of research on nucleic acid vaccines, in particular messenger RNA. You may have heard of those. The two leading vaccines that came out first from Pfizer and Moderna are based on messenger RNA, which is literally the molecule that is picked up by a ribosome and translate protein. So it's literally, let's just go to the message, put the message into cells, and let those cells in an individual who's been vaccinated express the protein to induce the immune response. A number of things about that. I'm surprised at how effective it's been, because it really does stimulate a really good response. But the rapidity with which you can make a vaccine like that, I mean, we have to go through a fair bit of work to generate a system where we can make our protein, our grams or kilograms of protein. With the genetic message, you just make the RNA. And if you can turn that around relatively quickly and use that as a vaccine, and that's really been shown to be extremely effective. So a wide range of approaches to making the vaccines. The last count, I think there were 70 clinical trials underway with 70 different vaccines. Now that you might not have the experience to grasp the significance of that, but at any one time prior to this last year, you'd not have one or more than one or two different vaccine approaches for a particular target going on at any one time. This has been an amazing year.
But I also remember talking to experts a year ago, and they were saying, we don't know if it's even possible to create a vaccine against a coronavirus, and here we are with 70 potential options. It's insane.
It is crazy. I mean, it turned out that it was a tractable target. I mean, people have been working on an HIV vaccine for 30 or 40 years, and we're still not there with that one. This is a virus that has a fairly obvious target, the spike protein. If you induce antibody responses to that spike in the right sort of way, you will afford protection. So as long as you can deliver it appropriately, and we've now worked out a number of ways of doing it, that's going to work.
How long does it normally take? I think it's good for us to kind of bear in mind how long it normally takes to make a vaccine and why it's been possible to make these coronavirus vaccines in such a shorter amount of time.
So the typical timeline for developing a vaccine from concept through to getting it commercialised and out there into the community is about 15 to 20 years. The fastest that anyone's ever gone is 4 years, which was really remarkable.
And this has been like 1 year. So
the first vaccine, Pfizer, went into emergency use after 11 months from the date that the sequence was first shown, and then anyone could start working on it. So it has been remarkable. I mean, I'll be honest, one of the biggest reasons is money. So if you throw enough money at this problem and get enough people working on it, and literally billions of dollars have been thrown at this, then you're able to scale do experiments at scale and progress it. I mean, I've been working on vaccines for many years, and you write a grant proposal, you wait a year to see whether it's been successful, you do that work, you write another grant proposal. So that's one of the reasons why discovery takes quite a long time. You've then got to interest a commercial company into taking it on board. Phase 3 clinical studies, I mean, one thing that people have been a bit concerned about is that we've done it so fast that we've cut corners. We haven't. It's just been remarkable what we've been able to do with the funding, so we've been able to parallel a lot of activities and do it at risk. No one would ever have thought of making large-scale vaccines before you've proven it works. And we did that this year. We actually made millions of doses before we got to the point of licensing. So paralleling those activities was one way. But it really is just the concerted global effort into and the sharing of data that has allowed us to move as quickly as we have.
It is pretty insane to think about the fact that that could possibly have worked for other diseases that we've been living with for a really long time. Like, COVID's a problem for the whole world's banded together, they put all their money into the same pot, and they've effectively nearly solved this problem. Should we be taking this approach for other diseases that we're working with?
I think there's a lot of view out in the community, the scientific community working in vaccines, that we've learned a lot from this. And the regulatory pathways, the timelines we'd previously considered were acceptable, I think in the future will be unacceptable. And we will probably, I don't think we'll ever be able to do it quite like this again, unless there's another pandemic, because of the imperative and the urgency of developing this. But I think we have learnt enough for us to probably shorten that timeline to subsequent discovery. And as I said, the really big news out of this last year is that messenger RNA technology, which can be applied to a wide range of different targets.
So Larissa, how long until we're all vaccinated? What does the rollout process look like?
Yeah, it's a really good question. And it's pretty complicated, right? I mean, we have to, with the different vaccines, they have different requirements. So we know for the mRNA vaccine, so that's for us here in Australia, the Pfizer vaccine, they have to be kept really cold. And that makes it hard to get to sort of more regional locations. Certainly the AstraZeneca, so the vector vaccines, they can be kept a little bit warmer, which makes it a lot easier to transport it around. The recombinant protein approaches, which is what the UQ vaccine was, also they're much easier. Can they be stored at room temperature even? Yeah. So they're more stable. So that's a really important consideration. There's also the case of there's manufacture. So now that CSL is able to manufacture the AstraZeneca vaccine onshore, it means that we can roll out things much more quickly, but it's also the distribution. How do we get everyone enrolled in time? How do we get enough doses to different GPs? This is all stuff that has to be worked out. So, you know, it's not a smooth process, but we're also in a privileged position here in Australia, right, where we don't have local community transmission. We want to have everyone vaccinated as soon as possible, because then it's much more likely that we'll be safe if we do open up the borders again. And we want everyone to be vaccinated around the world, because the more people that are vaccinated, the less opportunity the virus has to replicate. So one of the things that Paul mentioned was that these viruses, they have a natural sort of mutation rate. If you imagine every time the virus replicates, it's like it's, you know, copying a page in a book, and every time it copies it, it introduces one mutation, so one like small typo, if you imagine. And in that one first copy, maybe it's just one word that has one letter changed, and you still can read it, doesn't make much of a difference. But if you imagine that being copied over and over again, suddenly a whole word changes, and then suddenly a whole sentence changes, and then you have a totally different meaning of a word. And for a virus, it's kind of the same. So these mutations accumulate naturally, but when we have immunity, then there's selection of the fittest virus, so the one that kind of survives the best under that immune pressure. And when you have the virus replicating in so many different people, and then potentially in people who have weakened immune systems, so this is particularly people with HIV, so South Africa is a place where this happens, then you've got a lot more chance that the virus is going to change and accumulate mutations in places that it can evade our immunity. And this is where we've started to see the emergence of these variants. So I'm sure you've all kind of heard about the UK variant, the South African variant, there's a Brazilian variant. And we think that potentially they're more transmissible, so they're able to bind to and get into our cells better, but potentially they're also able to escape from our immunity better. And that's something that scientists are still kind of trying to work out. And that in itself could be a problem for future vaccines. But the sooner we can vaccinate everyone, the sooner we reduce the chance of those variants emerging. So we really need to vaccinate as many people as possible, as soon as possible, not just in Australia, but worldwide.
Yeah, I think just to add to that, some of the other things that are coming out, even though we're all still focused on COVID in context of trying to we're developing a new vaccine that will hopefully roll out at some future date, but we're also trying to understand the virus a bit better. We're trying to understand how to control it, to treat the disease, all of those things. But at the same time, we're looking forward. And there are a lot of lessons that have been learnt locally, not just at a global scale from a scientific perspective. But one of the emerging concerns we had last year when our borders were locked and we couldn't get anything in, we started to think about, well, what is our sovereign capacity? What is our capacity just here in Australia to do some of these things? From our perspective with our vaccine, we were very fortunate in that the one and only manufacturer in this country, CSL, was capable of making our vaccine. But they're not capable of making the RNA vaccine. They've had to tool up to make the vectored vaccine, and there'll probably be other vectored vaccines they won't be able to make. So there have been a lot of what we call sovereign gaps identified. And so the learnings we're taking out of this, we're already thinking of any future pandemic, and there will be another one, that we will be better placed to actually handle that by putting in place some of the bridges to these gaps.
So you mentioned that another pandemic's inevitable. You mentioned it as well, Larissa. I read the fine print, it's obviously coming. And Paul, your vaccine technology that you developed at UQ was originally for disease X, like for a virus or a disease that we knew would probably pop up at some stage, but we didn't know what it was going to be yet. Turns out it was COVID, but this isn't the last time we're going to see a new pathogen. It's not a question of if, but when. So like, when? When are we going to see another pandemic? How do we prepare for this?
It's a good question.
Would you like the crystal ball?
Yeah, exactly. I really, really would. I think people argue for a One Health approach. So, a lot of these pandemics, we know they come from crossover from other animal species. So it's important to study viruses in other animal species, and kind of one of the big contenders for that are bats. So bats are really unusual in that they have this kind of crazy immune system that we don't understand, which means that they harbour a lot of viruses, but they don't get sick from them. And we still don't really understand that properly. So probably understanding a lot more about what viruses are in bats and how bats control that will be really useful to know. Also, this One Health approach, particularly in the flu field, there's a lot of concern about avian influenza, so bird flu, obviously flu in pigs, and how do we control that? So things we can do are to try and develop broad-acting antivirals, so kind of like we've got broad-spectrum antibiotics. The more we understand about lots of different viruses, the more we can say, well, what is something that happens between, that's common to all viruses, and how can we target it? And people are definitely trying to look at that, right? So that would be something we'd have on hand for the next pandemic and say, okay, this is what we can use. Another thing that we sort of try and do is to treat the symptoms. So, for me, we're looking at when the immune system gets too overactive, how can we treat that and calm that down? And how can we boost people's immunity? So for COVID, of course, we know that of all the people who catch COVID, only a certain percentage get sick. So why is that, and what do the people who don't get sick have, and what are they doing, and what is their immune system doing right that we can boost in the other people? And similarly, what can we calm down? What this has really taught us, this whole pandemic, is just how little we knew about coronaviruses before this came up, because I'm sure Paul can allude to this. The common coronaviruses basically caused a common cold, so you can't get funding to study that. People like way more important diseases. The other ones were SARS and MERS, which you need special PC3 facilities to work in, which are expensive to run.
That's
you need a lot of PPE, a high containment facility. And now that we've got a pandemic worldwide, like certainly at UQ, we've upgraded our PC3 facilities. I'm in there all the time working with coronaviruses. A lot of people are interested in these things now. So we have gained a lot just in understanding fundamental disease, and that basic research is incredibly important for fighting off the next pandemic, you know?
Yeah, it's sad to think we needed this, but I think one of the consequences of having gone through this pandemic and the huge strain it's taken on many countries, not just in terms of individuals who've died, particularly overseas, but also our own citizens. But the economic impacts have been huge, and I think that's telling our decision makers that they need to take this a bit more seriously and invest in preparedness strategies, which we've been trying to get across for 20 or more years, but mostly into deaf ears because it wasn't a priority. And I think now that we've seen the consequences of what can happen with an emerging outbreak like this, that more efforts will be put into this preparedness, you know, the broad spectrum antivirals, which they've been generated in labs, but you just can't get them out. There's what they call this translational valley of death. So the early discovery science, and then there is the commercial development, which costs a lot of money to do and requires the investment of big pharma. So this valley of death is trying to get through with taking a good idea through into translation. And what we need to be doing is filling that, bridging across that valley of death with appropriate resourcing. You know, the manufacturing capacity to generate material for clinical trials, for example, that will then attract a big pharma to take it on board. So, you know, the imperative to do that, say for developing a broad spectrum antiviral against coronaviruses, people had spoken about it. I've been to meetings where that was discussed 5 years ago, but no one took it seriously. I think we're in that a different situation now, where I think we will get the funding and the resources to actually take us through to that better prepared state.
So it's come at a high cost, but hopefully the things that we've learned from the coronavirus pandemic will assist us in our next race for a cure. Thank you so much for coming to join us today. Please thank Larissa and Paul for their expertise. Thank you.